Introduction. The cell was first seen by Robert Hooke in 1665 using a primitive, compound microscope. He observed very thin slices of cork and saw a multitude of tiny structures that he resembled to walled compartments of a monk. Hence, named them cells. Hooke's description of these cells was published in Micrographia. The cell is smallest unit of a living system and fall in the microscopic range of 1 to 100 micrometers. They attain various shapes and sizes to attain variety of functions. The understanding of cell is necessary to understand the structure and function of a living organism. One of most important characteristics of cell is ability to divide. The existence of a cell indicates that it has evolved from an already existing cell, and further it can give rise to a new cell. This was first stated by Theodor Schwann. Pioneering work by Theodor Schwann, Matthias Jakob Schleiden on cells, gave birth to the cell theory. Their theory states: 1. All living things are made of cells. 2. Cells are the basic building units of life. 3. New cells are created by old cells dividing into two. In 1855, Rudolf Virchow added another point to the theory and concluded that all cells come from pre-existing cells, thus completing the classical cell theory. The cell theory holds true for all living things, no matter how big or small, or how simple or complex. Viruses are exception to the cell theory. Cells are common to all living beings, and provide information about all forms of life. Because all cells come from existing cells, scientists can study cells to learn about growth, reproduction, and all other functions that living things perform. By learning about cells and how they function, we can learn about all types of living things. Classification of cells. All living organisms, bacteria, blue-green algae, plants and animals, have cellular organization and may contain one or many cells. The organisms with only one cell in their body are called unicellular organisms: bacteria, blue-green algae, some algae, protozoa, etc. The organisms having many cells in their body are called multicellular organisms: fungi, most plants and animals. Any living organism may contain only one type of cell, either prokaryotic cells or eukaryotic cells. The terms prokaryotic and eukaryotic were suggested by Hans Riss in the 1960s. This classification is based on their complexity. Further, based on the kingdom into which they may fall, i.e. the plant or the animal kingdom, plant and animal cells bear many differences. Prokaryote means before nucleus in Greek. They include all cells which lack nucleus and other membrane-bound organelles. Mycoplasma, virus, bacteria and cyanobacteria or blue-green algae are prokaryotes. Most prokaryotes range between 1 micrometer to 10 micrometers, but they can vary in size from 0.2 micrometers to 750 micrometers. Thiomargarita namibiensis. They belong to two taxonomic domains, which are the bacteria and the archaea. Most prokaryotes are unicellular, exceptions being myxobacteria, which have multicellular stages in their life cycles. They are membrane-bound, mostly unicellular organisms lacking any internal membrane-bound organelles. Though prokaryotes lack cell organelles, they harbor few internal structures, such as the cytoskeletons, ribosomes, which translate mRNA to proteins. Membranous organelles are known in some groups of prokaryotes, such as vacuoles or membrane systems devoted to special metabolic properties, e.g., photosynthesis or chemolithotrophy. In addition, some species also contain protein-enclosed microcompartments, which have distinct physiological roles: carboxysomes or gas vacuoles. Cell wall. Cell wall is the outermost layer of most cells that protects the bacterial cell and gives it shape. One exception is mycoplasma, which lacks cell wall. Bacterial cell walls are made of peptidoglycan, which is made from polysaccharide chains cross-linked by unusual peptides containing D amino acids. Bacterial cell walls are different from the cell walls of plants and fungi, which are made of cellulose and chitin, respectively. The cell wall of bacteria is also distinct from that of archaea, which do not contain peptidoglycan. The cell wall is essential to the survival of many bacteria. The antibiotic penicillin is able to kill bacteria by preventing the cross-linking of peptidoglycan, and this causes the cell wall to weaken and lyse. Lysozyme enzyme can also damage bacterial cell walls. There are, broadly speaking, two different types of cell wall in bacteria, called gram-positive and gram-negative, Figure 2. The names originate from the reaction of cells to the Gram stain, a test long employed for the classification of bacterial species. Gram-positive bacteria possess a thick cell wall containing many layers of peptidoglycan and teichoic acids. In contrast, gram-negative bacteria have a relatively thin cell wall consisting of a few layers of peptidoglycan surrounded by a second lipid membrane containing lipopolysaccharides and lipoproteins. These differences in structure can produce differences in property as antibiotic susceptibility. For example, vancomycin can kill only gram-positive bacteria and is ineffective against gram-negative pathogens, such as Pseudomonas aeruginosa or Haemophilus influenzae. Morphology of prokaryotic cells. Prokaryotic cells have various shapes. The four basic shapes are: Figure 3. Cocci, spherical. Bacilli, rod-shaped. Spirocate, spiral-shaped. Vibrio, comma-shaped. Eukaryote. A eukaryotic cell consists of membrane-bound organelles. They belong to the taxa Eukaryota. All species of large complex organisms are eukaryotes, including animals, plants and fungi, and most species of protist microorganisms. Eukaryotes appear to be monophyletic, organisms that form a clade, and make up one of the three domains of life. The two other domains, bacteria and archaea, are prokaryotes and have none of the above features. Eukaryotes represent a tiny minority of all living things. Even in a human body, there are 10 times more microbes than human cells. However, due to their much larger size, their collective worldwide biomass is estimated at about equal to that of prokaryotes. Unlike prokaryotes, eukaryotic genome is enclosed in the nucleus surrounded by the nuclear membrane. Other than the nucleus, many membrane-bound organelles dwell in their cell cytoplasm. Cell division involves separating of the genome, which is in the form of tightly packed condensed structure known as the chromosomes, through movements directed by the cytoskeleton. Classification. The eukaryotes are composed of 4 kingdoms: Kingdom Protista, Kingdom Fungi, Kingdom Plantae, Kingdom Animalia. Cell features. Eukaryotic cells are much larger than prokaryotic cells. Range between 10 to 100 micrometers. They have a variety of internal membranes and structures, called organelles, and a cytoskeleton composed of microtubules, microfilaments, and intermediate filaments, which play an important role in defining the cell's organization and shape. Eukaryotic DNA is divided into several linear bundles called chromosomes, which are separated by a microtubular spindle during nuclear division. Internal membrane. Eukaryote cells include a variety of membrane-bound structures, collectively referred to as the endomembrane system, involved in various functions. Simple compartments, called vesicles or vacuoles, can form by budding off other membranes. Many cells ingest food and other materials through a process of endocytosis, where the outer membrane invaginates and then pinches off to form a vesicle. It is probable that most other membrane-bound organelles are ultimately derived from such vesicles. The nucleus is surrounded by a double membrane, commonly referred to as a nuclear envelope, with pores that allow material to move in and out. Various tube and sheet-like extensions of the nuclear membrane form what is called the endoplasmic reticulum or which is involved in protein transport and maturation. It includes the rough where ribosomes are attached to synthesize proteins, which enter the interior space or lumen. Subsequently, they generally enter vesicles, which bud off from the smooth In most eukaryotes, these protein-carrying vesicles are released and further modified in stacks of flattened vesicles, called Golgi bodies or dictyosomes. Vesicles may be specialized for various purposes. For instance, lysosomes contain enzymes that break down the contents of food vacuoles, and peroxisomes are used to break down peroxide, which is toxic otherwise. Many protozoa have contractile vacuoles, which collect and expel excess water, and extrusomes, which expel material used to deflect predators or capture prey. In multicellular organisms, hormones are often produced in vesicles. In higher plants, most of a cell's volume is taken up by a central vacuole, which primarily maintains its osmotic pressure.